Hello, I'm Grace Hobson. I'm a principal research scientist emeritus at Nemours Alfred I. DuPont Hospital for Children. I'm retired now, but I maintain an affiliation with Nemours so that I can keep doing a little work. As some of you are aware, my lab was among the first to develop molecular diagnostics for PMD in the 90s. Today, I'm going to tell you the story of one of the projects in my lab that I started with finding an interesting mutation in a patient back in the early days, and I hope this story is not finished yet. This is a brief introduction to the key players in PMD. Here, indicated in red, the diagram shows an oligodendrocyte cell in blue, wrapping its cell membrane around the axon of a nerve cell in red to make the myelin sheath, which insulates the axons so that the nerve impulses travel properly, similar to the function of insulation on electrical wires. When the PLP protein in the myelin sheath is defective because of a mutation in the PLP gene, the nerve impulses are not transported properly throughout the central nervous system. This is some of the key players in the genetic code. You've probably heard that the affected gene in PMD is the PLP1 gene, and it's in the DNA in the nucleus of the cell. It codes for a protein called proteolipid protein, or PLP1. What you may not have heard about is the RNA, shown as a single strand in red in between the double-stranded DNA and the protein in this cell diagram. RNA is important because it's made using the code in the DNA template, and it carries the code out of the nucleus to where the protein is made. At the bottom of the diagram, you can see the protein-making machinery sitting on the red strand of RNA, reading the code, and adding the yellow beads of amino acids to the protein. The RNA that originally gets made from the DNA template has to get spliced before it's read to make protein. The boxes in this stick figure represent the regions of RNA that contain the code for protein, called exons. The regions between the exons that don't code for protein are introns, and they have to be cut out of the RNA by the splicing machinery of the cell. At the bottom, during splicing, the exons have been brought together into what's called the messenger RNA, or mRNA, that codes for the protein. In the PLP1 gene, there's an interesting alternative splicing event that occurs between exons 3 and 4. There are two spliced donor sites at exon 3 that can be used to join the RNA to exon 4. The splice that makes an mRNA form called DM20, shown at the bottom on the left, doesn't have the little block labeled 3B, while the form that's called PLP1 on the right does have the 3B block. This is a diagram of the PLP1 protein in the myelin membrane. The 35 circles in blue are the amino acids for the 3B block that's not present in the DM20 spliced form of the protein. The relative amount of the DM20 and PLP1 forms made in oligodendrocytes is tightly controlled in cells. Before myelination occurs, DM20 is the only form, and it's found at low levels. As myelination begins, PLP1 turns on, and there's a large increase in the amount of both forms. After the peak of myelination, PLP1 is the predominant form, and that continues throughout adulthood. About 70% of the total is PLP1 form, and 30% is DM20. We found that a number of the mutations shown at the asterisks in this figure were located near the alternative splice region. The ones that don't make an amino acid change are in red. These patients didn't have any other mutations in the PLP1 gene, so we wondered if these mutations could be affecting the relative levels of PLP1 and DM20 forms produced by the alternative splice. And if so, maybe these patients had disease because they have abnormal levels of the alternatively spliced forms, rather than because of making a mutant protein. To determine whether the patient mutations affect relative levels of PLP1 and DM20 forms, we first made an artificial DNA construct each with separate patient mutations. At number 1, I've shown the construct with patient mutations named on it rather than just indicated by an asterisk. Then we put constructs on oligodendrocytes growing in culture dishes, and they take up these DNA constructs. They make RNA from them and splice the RNA. Then we prepare the RNA from the cells and perform an assay to determine relative levels of PLP1 and DM20 forms that are made. This is our results for an experiment testing 9 mutant constructs. The normal construct is the first bar on the left, and you see that most of the patient mutations either reduce or eliminate the PLP form from what's seen in normal. So they are affecting alternative splicing. Then we wanted to make a mouse model to establish that a mutation affecting the PLP1 and DM20 alternative splicing was disease-causing. And I'd like to call your attention to one of the mutations that we call DEL, marked in red on the stick figure and the graph. This mutation is the one that we chose for making the mouse. The patient with this DEL mutation had an unusual clinical presentation that was milder for PMD than most, but more progressive over time. It was one of the early mutations we identified in a patient back in the 90s. It's a 19-base deletion of coding sequences in a region that doesn't code for protein. But we showed that the specific bases had to be there for the alternative splice to work properly. So when they're deleted, it doesn't work properly. Later, we showed with Nicole Wolf's group that patients with mutations that decrease PLP1 spliced form have a clinical presentation that's a bit different from typical PMD. And these patients had what she named hypomyelination of early myelinating structures, or HEMS, that was seen on MRI. Although we were not able to reevaluate our patient with the DEL mutation for HEMS on an appropriate MRI series, that is what our patient with this mutation probably has. This shows our results with the DEL mouse model. Very briefly, in A, we confirmed that the PLP form is decreased in RNA from the brains of the DEL mice from about 65 to 70% of total in wild-type mice to about 20% in DEL mice. In B, we showed by electron microscopy that myelin stability and compaction were affected by the mouse. And in C, in brain sections for inflammatory markers, we showed that there was an inflammatory response to the myelin breakdown. These data from the papers provide the connection between the splicing defect and disease. Antisense oligos bind to single-stranded RNA, and we thought if we could make an antisense oligo to bind specifically at the DM20 splice site of the RNA, we could get alternative splicing to shift back toward a more normal level of the PLP1 form in cell culture and in our DEL mouse. So what's an antisense oligo? A strand of genetic code can be designed that is the antisense, as shown here in green, to the sense strand of the RNA, shown here in red, because A's in the code always bind U's and G's always bind C's. If you know the sense strand, then you can design the antisense strand using bases that will bind to it. It's called an oligo because oligo means few, and it's made of a few letters of the genetic code. Here we see an antisense oligo, or ASO, binding the RNA at the DM20 splice site. We call the specific antisense oligo that we designed ASO-PLP. ASOs can be made of RNA, but RNA is not very stable, so we used a type of synthetic oligo called a morpholino oligo that was designed to be more stable than RNA and hang around in the cells longer. Morpholino oligos are similar in structure to RNAs. We then needed to check that the ASO-PLP would increase the PLP1 form in cell culture. Here, like previously, we put these mutant constructs on the oligodendrocytes and they would take it up. But this time we either treated or not with ASO-PLP. In B, most of the constructs, including the normal one, which is first on the left, shifted alternative splicing significantly toward more PLP1 form when treated with the ASO-PLP at the green bars than when not treated at the black bars. The only two mutant constructs that didn't produce more PLP1 were the two shown in blue. These mutations were known to actually destroy the PLP1 splice site. So even if you block the DM20 splice with the ASO, you can't get it to produce any PLP form. So now we have an ASO that shifts splicing toward PLP form for most of our mutants. But we needed to make one more thing before testing in mice, and that is a random control ASO, like the one shown here in blue, that won't bind to the RNA at the splice site because it's not the antisense of the coding sequence for that RNA. Here, I've just picked a code sequence at random to show us an ASO in blue that won't bind, but we actually used a mixture of ASOs with random bases at each position. This is a diagram of the scheme for our mouse treatment experiment. On postnatal days 1 or 2, and postnatal is days after birth, we injected ASO-PLP or the random control ASO at one of two amounts into the ventricles of brains of mice. The ventricle is just a space in the brain. And then at postnatal days 21, 60, and 90, we euthanized groups of the mice and prepared RNA from half brain and half the spinal cord and made protein extracts from the other half, and then performed assays to detect PLP1 RNA and protein. This is our data from treating the DEL mice with ASOs presented in 3 graphs. In A, we're looking at the results in RNA prepared from the brain. Treatment of DEL mice with the random control ASO at the blue label did not change PLP1 levels from untreated DEL mice at the red label at any of the 3 time points, which are indicated by open bar, gray bar, and black bar. But the treatment with both amounts of ASO-PLP at the green label increased the PLP RNA form closer to levels in the normal mice, and the effect was sustained to 90 days. We saw basically the same result in B when we looked in spinal cord RNA, and basically the same result in C when we looked at the PLP protein in brain. So our data show that treatment of DEL mice with ASO-PLP brings brain and spinal cord levels of the PLP1 form of RNA and protein closer to normal, and the effect is sustained to 90 days, while the random control does nothing. So we conclude that the treatment with our ASO-PLP corrects alternative splicing of PLP1 in the mutant DEL mouse. But then the question is, did it correct the disease process? I showed you on a previous slide that the myelin instability in the DEL mouse causes an inflammatory process in the brain. Here we looked at the levels of 4 markers that are increased during inflammatory processes to see if we could see improvement in our treated mice. Again, here, the patterns of the 4 graphs are very similar. The first 2 bars of each graph tells you that the amount of inflammatory marker that you're looking at in a particular graph increases in the untreated DEL mice from day 60 to day 90. So inflammation is progressive when untreated. The next 3 bars are data from treatment with the random control at the blue labels. Here we would hope that levels were not changed, but the ASO or the injection itself seems to have been affecting some of the levels a bit. However, these these effects were very small when compared with treating with the ASO PLP at the green labels in the last 3 bars, where significant reductions in the 4 inflammatory markers were seen. And although we treated only once at day 1 or day 2, the improvement was sustained out to 90 days or 3 months. So we conclude that treatment with ASO-PLP not only corrected alternative splicing of PLP1, but it also improved inflammation in our mouse model, indicating that ASOs have therapeutic potential for treating PMD. This is a summary slide. I've shown you that when tested in culture, some PMD patient mutations cause a decrease in the PLP alternatively spliced form, and treatment of the cultures with ASO-PLP can increase the PLP form. We made a mouse model of one of these patient mutations called the DEL mutation. The PLP1 alternatively spliced form was decreased in the mouse. The mouse also had myelin instability and brain inflammation, suggesting that the alternative splicing mutation was disease-causing. Treatment of the DEL mice with ASO-PLP produced sustained correction of the alternative splicing defect at both the RNA and protein levels, and it improved the brain inflammation, suggesting that ASOs have therapeutic potential for treatment of patients with defective alternative splicing. The focus today is on therapeutics that will generally be applicable for the largest numbers of patients, rather than a small group like those with defective alternative splicing of PLP. However, to make the most significant improvements for some patients, it'll be important to design therapeutics to attack the specific molecular defects in a group of patients, or even in individual patients. And my hope is that the PMD patients with alternative splicing defects will be among the first for whom these more personalized therapeutics are made. I would like to acknowledge the people who did the work in my lab and the support that I received over the years to do the work. Stephanie, who did most of the mouse work, Karen, a longtime research associate who still helps me from time to time, postdoc Jen, and Caitlin, who's now a neonatologist at Christiana Care. Sadly, Stephanie and Jen both passed away since this work was done.